To begin our presentation in this study and finding, entitled Correlation of Eksotika Malaysia Papaya with Genetics Modified Fruits, let's check out our introduction. Papaya, or its scientific name, Eksotika papaya, is an economically important fruit in many tropical and subtropical countries. Besides providing food to the people, the crop has potential to be exploited as an income generator in many countries including Malaysia. Through the past years, papaya have had some trouble where they were very inconsistent in yield and generally had very poor eating qualities. Hence, to overcome these issues, Malaysia Agricultural Research and Development Institute (MARDI) have developed an Eksotika Malaysia Papaya, or its scientific name, Malaysian Eksotika Papaya L. varians Eksotika, in 1987. There are three objectives in this study and finding, which are to explain the genetic engineering process of Eksotika Malaysia Papaya as genetic modified organisms, specifically fruit. Next, to justify the breeding method of Eksotika Malaysia Papaya and to identify the advantages, disadvantages, and ethical issue in genetic modified organisms, specifically fruit.
For the methodology part, as you can see in the figure 1, the sunrise swallow who have sweet flesh and sweet smelling but small in size, meanwhile Subang 6 who have less sweet flesh but bigger in size, make an Eksotika Malaysia Papaya that have a sweet flesh and big fruit through the back cross breeding.
Results show the comparison between transgenic papaya, which is Eksotika papaya as an example, and non-transgenic papaya during E. mallotiflora infection, or known as papaya diabetes. This infection has bring a serious problem to the non-transgenic papaya. It makes the papaya to yellowing and necrosis along leaf ends, followed by water soaked areas on the base of leaf stalks, crowns, and along leaf mid ribs. To know more about the transgenic papaya, we will move to the discussion. Genetic engineering process used to produce transgenic papaya that are resistant to E. mallotiflora infection, and acyl homoserine lactone AHL lactones sp24 and acyl homoserine lactones CHB18 genes, which have high quorum quenching potential against E. mallotiflora, have been isolated and transformed by using the agrobacterium-mediated transformation method into embryonic callus of Eksotika Malaysia Papaya. Next, the genetic engineering process used to produce Eksotika Malaysia Papaya that has a longer shelf life. The 1-aminocyclopropane 1-carboxylic acid oxidase 2 SCO2 genes is manipulated, so the production of ethylene, which leads to the ripening in the Eksotika Malaysia Papaya, can be reduced by using a process called antisane technique. This transgenic fruit can delay the ripening up to 15 days compared with 4 days of non-transformed seed-derived papaya fruit.
Under the process of breeding, simple mass selection can be used where the best trees for seed propagation was picked. Backcross breeding is also one of the ways to breed the papaya, as we can see in figure 1, where the single character from the non-recurrent donor parent will be transferred to the recurrent parent. As an illustration, large fruit owned by Subang 6 will act as the non-recurrent parent, while sunrise swallow who own a superb eating qualities will act as the recurrent parent.
To elaborate further, there are also advantages of this transgenic papaya, which are it is a good bearer plant and have aesthetics qualities. This actively demonstrates that compared to the non-transgenic papaya, this plant can achieve food production about 60 tons per harvested area per year. On top of that, it has an orange-red flesh with pleasant aroma and has contributed to become the main variety grown for commercial and export purpose and directly able to boost the agricultural economy. Along with this, there are some disadvantages arising in the development of Eksotika Malaysia Papaya, which are Eksotika disease and high sensitivity to low temperature. According to the Journal of Eksotika Malaysia Papaya Current Research Strategies Planting Challenges, published on 2018, Eksotika disease happen due to pests such as fruit flies and spider mite aiming at the papaya foliage, fruits, and roots. It is mainly a microorganisms related disease. Moreover, the sensitivity to low temperatures will cause chilling injury and rapid deterioration of the fruit quality.
So for the ethical issues, according to the Food and Drug Administration (FDA), the health effects of food grown from genetically engineered crops depend on the composition of the food. Any new product may have either beneficial or occasional harmful effects on human health. With the understanding of science and modern set of experience with biotech-derived plants, they appear to be safe. The consumers find biotechnology acceptable when they believe it offers benefits and it is safe. To sum up, Eksotika Malaysia Papaya, also known as Ekro Solo, was developed by Malaysia Agriculture Research and Development Institute by crossing Sunrise Solo Papaya and Subang 6 Papaya, which is a great advancement in agriculture field. Genetic engineering used to create Eksotika Papaya resistant to dieback disease, known as transgenic papaya, which is caused by Erwinia mallotivora. Eksotika Papaya with a longer shelf life was also developed using genetic engineering. There are many methods such as mass selection and back crossbreeding to breed papaya by conventional breeding. Plus, it is a strong beer plant and has aesthetic qualities that assist in the purposes of trade and export. However, this transgenic papaya has the advantages such as exotica disease, which is caused by pests. It can be inferred that biotechnology is important for improving fruit quality. All in all, the objectives of this study and finding have been achieved successfully.